This is a production of Cornell University.
Thank you for the very nice introduction, Bruce. Just a quick seminar overview. First, going to talk about disease resistance breeding in the Cornell grape breeding program. Then I'm going to talk about three topics that made up my dissertation. The first is using computer vision to detect or quantify Downy mildew and leaf trichomes. The next part will be to integrate genetic information, SNPs in this case, with multiple phenotypes in something called a Bayesian network. And finally, I'll talk about accounting for spatial effects in field data with grapevines. So just an overview of grapes. They're perennial, clonally propagated. There are two main groups that we work with: Vitis vinifera, which is the European grapevine. It's susceptible to virtually every disease, but it has very good wine quality as well as certain table grape attributes that we want. And the other group is various Vitis species that are native to North America. And so these species are resistant to nearly every disease. However, they have really poor wine quality. However, sometimes we do want their table grape characteristics. And so the goal of the program is to merge the best of both worlds to obtain hybrids that have a high disease resistance as well as high wine quality or table grape characteristics. So how grape breeding works in the Cornell make process, we terminate seed. Then if we have markers, we have certain markers for traits such as disease resistance, seedless, that we have obtained from F1 families using QTL analysis of F1 families. Then those seedlings are transplanted into the vineyard. And, after years of multi-location, multi-year trials of evaluating for field disease resistance as well as wine quality, the best of those selections are released as cultivars. So the first part I'll be talking about is computer vision. So what is computer vision? It's using algorithms to extract information that's from images that's usually extracted using human vision. And so the pros is that this method is high throughput. You get a lot of data fast. That you could obtain quantitative phenotypes, and that there's a lot of high-level programming libraries that are available in order to implement these algorithms. For example, the one I used is called OpenCV or Open Computer Vision. Some negatives with this method is that it's time-consuming to develop, and it requires technical knowledge from the end users, which the phytopathologists or breeders in this case. So a little about Downy mildew. This is how it looks like. So on the left-hand side is the abaxial side of the leaf, and this is where the oomycete infects through the stomata, and you obtain sporulation. And essentially, for Vitis vinifera, you need to spray about every 2 weeks if it's a really rainy year. And so it's commonly thought that this kind of intensive pesticide application is no longer sustainable. So now I'll talk about the genetic resources that I've used to evaluate this method. The first is, or both of them are interspecific F1 hybrids. The first is Vitis rupestris bihorizon. So Vitis rupestris is a Native American Vitis species. And horizon is an interspecific cross. It has Vitis vinifera in its background, as well as other Vitis species. And so for both of these, we have genetic maps available. So there's about 6,000 SNPs, and the population size is about 160. So I'll call this family RH. And the other family is Horizon by Vitis cinerea. Again, Vitis cinerea is a wild Vitis species. And what's special about this family is that it has a baxial leaf trichomes. So there's 2 images of leaf discs per family. The left-hand one is prior to inoculation, and on the right-hand side is the leaf disc that what it looks like, the same leaf disc after inoculation. And so you could see both of them, the sporulation on both, but it's much harder to see on the leaf disc when it has leaf trichomes, and that's because the leaf trichomes are white and the sporulation is white, so it's difficult to differentiate them. And so the idea that I'll use is using the computer vision rating prior to inoculation as a covariate to control for the leaf trichomes. And so the particular assay that we use is a leaf disc assay. So basically, we took leaf discs, or took leaves from the field, made leaf discs, and plated them on an agar dish, and the leaf discs corresponded to genotypes shown in the grid here. The experimental design here is for the RH family. We had controls, and we did this over 2 years, 2015 and 2016. In 2015, we had 3 experiments. In 2016, we had 2. For each experiment, there was a time series collection of data from 3 DPI, or days post-inoculation, to 6 DPI, or sometimes it was 4 DPI to 7 DPI if we didn't see sporulation at 3 DPI. So we collected 4 leaves from each genotype, and each leaf, from each leaf we extracted 2 leaf discs. So for each experiment, we had about 1,400 leaf discs. And so the only difference between the RH family and the HC family experimental design is that in 2015, we only had 2 experiments for the HC family, and that was just due to lack of time. So we compared the computer vision rating with a manual rating. So this is the scale. It's a 1 to 5 scale, where 1 is no sporulation and 5 is dense sporulation. So how we did the computer vision, or what the algorithm did, is that you have this input image, and what we want to do is remove the background, the agar, from it, as well as detect the leaf discs. And so this requires some user input of parameters. And so the next thing that we want to do is, if you notice on the second row of the leaf disc, there's greater illumination than in the first row. And we had to correct for that. And did something called a Wallace filter, which I won't go into, but essentially corrects or normalizes the illumination across an image. And then we could detect our phenotype, which in this case is the amount of white pixels in a leaf disc divided by the area of a leaf disc. So now I'll go into the results. First, I'll just explain one experiment. So here is the computer vision ratings on the left-hand side. So this cyan color is 3 days post-inoculation. And so you could see it's very close to 0, as you would expect. And then 4 days, we get kind of the distribution moves up. And as you get further along in the days post-inoculation, the distribution keeps moving forward. And it's relatively normally distributed. However, compare this to the manual sporulation ratings. You could see at 3 dpi, it's skewed all the way to the 1 rating. And as you get further along, it skews towards the 5. And so the ratings here are the average of 8 leaf discs for a particular genotype for an experiment. So here are all the other distributions. And you could see that the trend is similar across experiments where it's relatively normally distributed in the computer vision ratings, but very skewed in the manual sporulation ratings. Looking at the correlations between methods, we see that the correlations are very high. And so this shows that our method, our computer vision method is accurate in obtaining a good phenotype. And you can see when we average across genotypes, the correlation increases, which is what we would expect. So now going on to the computer for the HC family. So this family had leaf trichomes. And so the computer vision ratings were the ratings after accounting for the leaf trichome computer vision ratings, which were simply the computer vision ratings at 2 days post-inoculation prior to any sporulation on the leaf discs. And so you could see that it's not as clear in the computer vision distributions, that there's a general trend of increasing percent sporulation. However, it's not as clearly defined. And for example, in the first experiment at 7 days post-inoculation, it kind of moves down instead of being close to 5 and 6, which are the blue and purple in the first graph. However, for the manual sporulation ratings are again skewed, or in the case of the first experiment, it's just uniformly distributed. Looking at the correlations, they were much lower. Averaging across genotypes helped increase the correlations a little bit, but not by a lot. And for one experiment, all the days post-inoculation correlations weren't significantly different than 0. And the reason for that is we didn't get a good rating of essentially the computer vision rating didn't do a good job in obtaining the leaf trichome, the true rating. And so kind of the use of the covariate didn't really work in that case. And so one way to improve it would be to standardize illumination to maybe improve obtaining that leaf trichome rating to use as a covariate. There's a summary looking at the rating and processing times over 1,400 leaf discs. The manual visual rating took 3 hours, while the computer vision method took 20 minutes. So there's a great improvement in how much you could phenotype. And if you want more information about this, the phytopathology paper is out on for this research, and all the code is on my GitHub in the link shown below. So some future directions in this research. There's a comment by Martin the last time I gave this talk about how if you see that because of this parameter search, kind of need to set parameters where you detect all the leaf disks at the same time, which is relatively easy to do for the RH family, but there are problems with it for the HC family, but you still get like detected circles that are much smaller in some cases. So this method that's recently been published, ELSDC, played around with it and seems like a good way to remove this parameter search. And another direction to go into is to generalize phenotype detection. For example, in my case, I'm detecting white pixels. However, what if the disease was red? If we could model the pixels as coming from particular clusters, then it could generalize this method. So reviewing the phenotypes we obtained for these two families, we have the manual and computer vision sporulation ratings. Going forward, I'll just talk about the manual sporulation ratings because of the kind of problems we've had in the HC family with these ratings. And we also, as I mentioned, collected the leaf trichome ratings at 2 days post-inoculation. And we also collected hypersensitive response, or RH, ratings, which were collected at 2 days post-inoculation before any sporulation. And what essentially the rating scale is the detection of black dots, which represent necrosis. So a rating of 5 has dense necrosis. And so one of the questions is, what phenotype should we use for QTL detection? We have 8 ratings per genotype per experiment. And how we integrate this is with a mixed model. So our response in this case is the average of 8 ratings. Our fixed effects are the intercept, the experiments within years, and the leaf trichome ratings, and the leaf trichome ratings only for the computer vision ratings for the HC family. Then we have a random effect of the breeding value, or the genotypes, which here we assume that all the genotypes are independent. And we include genotype by year effects, again, assuming that the years are independent. And we fit this using mRebel, which is an R package. So the simplest QTL detection is just using a single phenotype. So I'll use the term phenotype, but I'm really going to use the breeding value as the response variable in this case. So the basic idea is you scan the genetic map for significant SNPs, and you include them in your model, and you keep doing this until no more SNPs are significant. So for example, you have this equation where you end up with a particular phenotype equals an intercept plus SNP1 multiplied by the effect of SNP1 plus SNP33 multiplied by the effect of SNP33 plus the error term, where the SNPs are fixed effects. And we did this with RQTL, again, an R package. And one issue with this method is that the QTL are often not robust when you have a small population size. So in this case, we only have 160 individuals. So say you remove 10 or 10%, you might end up with different QTL. So we kind of want ideally a method that's provides robust QTL because when we're going to invest a lot of effort in using them for marker-assisted selection. So in order to essentially understand how kind of phenotypes affect phenotypes, how phenotypes affect, or how gene or SNPs affect multiple phenotypes. One thing we could do is adjust that prior method and simply scan for the most significant SNP or phenotype. And so you could end up with a model that's some phenotype 1 is the response and is explained by phenotype 2, for example. So in order to do this, we need to be sure that we include effects that only kind of make sense, make biological or mechanistic sense. So for example, if we have leaf trichomes equal or being affected by sporulation in this case, that doesn't make sense because leaf trichomes were present on the leaf prior to inoculation. And so that model will not make sense. And so we wouldn't consider it. And so an effective way to do that multiple phenotype analysis is by using something known as Bayesian networks. And so what you're doing here is you're adding effects in something called a greedy manner until an addition no longer increases the network's Bayesian information criterion, or BIC. So I'll explain a few terms in that sentence. The first is, what is a network? It's multiple separate linear models, one per phenotype. So these will just only be fixed effect linear models. The BIC is the likelihood of a particular model subtracted by a penalization term, which is a function of the population size and the number of parameters in that model. The network's BIC is simply a sum of the individual models, individual model BICs. And greedy addition means the addition of an effect to a particular model that increases the network's BIC the most. And so the reason they're called Bayesian networks is simply because we're using the Bayesian information criterion. So now I'll give an example of how this is done. So assume we have two phenotypes, sporulation and HR or hypersensitive response, and two SNPs. And so these orange arrows represent all possible effects. So immediately we'll not attempt to account for the effect of sporulation on HR because HR appears prior to sporulation, and we do not allow phenotypes to affect genotypes. So then we essentially go one by one with these effects and ask which one increases the network's BIC the greatest. And this is a hypothetical example, so it just SNP2 affects it the most. And we do this again, asking estimating every effect and asking which one increased the BIC the most. So it's this one in this case, then it's this one, and then we come up to a point where no addition of any of these arrows increases the BIC of the model. And so you're left with the two models which this network represents, which is that sporulation is affected by HR, which is this arrow, and SNP2, and HR is affected by SNP2 only. And so SNP2 has a pleiotropic effect affecting both sporulation and HR. So this method is too computationally intensive to do with thousands of SNPs. So we select a non-redundant set of SNPs for each phenotype. And it's, the name of that set is called the Markov blanket. So we'll have one Markov blanket for each phenotype. So then we construct 1,000 networks using random sets of 90% of the genotypes. We do this for both RH and HC families. Then we merge all the networks in one where we only keep the effects that appeared in at least 50% of the networks. And we do this with the bnlearn package in R. So the results for the RH family, so you could simply represent any linear model in this network representation. So I'll just do that for the single phenotype QTL analysis results. And so the difference only here is that the phenotypes will not affect each other in the single phenotype analysis. So what we see in the Bayesian network is that there's no effect of HR on sporulation, which is the only possible phenotype interaction that I allowed. And that the SNPs generally found are in the same chromosome, so the QTL are similar. However, the main difference is that in the single phenotype QTL analysis, the sporulation trait had many more QTL found for it than in the Bayesian network. So it's a little bit more interesting in the HC family, where we found that leaf trichomes have a negative effect, this red arrow, on sporulation and HR. And HR had a positive effect on sporulation. And so we don't know why this is the case, but for the leaf trichome negative effects on the disease phenotypes, it's hypothesized that it forms a physical barrier between the spores of the oomycete and the stomata on the abaxial side of the leaf. And that would prevent spores from germinating. And looking at the single phenotype QTL analysis, it's somewhat uninformative because all the phenotypes were explained by the same two QTL in chromosomes 5 and 8. So the question is, what's really going on there? And the Bayesian network kind of explains that. But otherwise, the QTL are similar, and you could see that 7 affecting sporulation is found here, and the QTL on chromosome 6 affecting HR is also found. So to summarize this, the disease resistance in the two families is controlled by multiple QTL, and they only account for a small % of the phenotypic variance. That breeding for leaf trichomes, as well as other QTL, could possibly lead to durable resistance, again, because you have that physiological type of physiological barrier mechanism, and say a mechanism that's biochemical, where there's, say, necrosis occurring to stop infection. Bayesian networks are one way to integrate multiple phenotypes with genetic data, as we've seen. And one question is, are Bayesian networks better models? So in order to do this, we would need to perform cross-validation. So I haven't done that formally, but preliminary experiments do suggest that Bayesian networks are better than just using single phenotypes. So now I'll move on to accounting for spatial effects in vineyards. So as I mentioned, we commonly phenotype in the field as opposed to in the lab, as the previous methods did. And it's commonly done in unreplicated vineyards, although not in all cases. So some assumptions one can make is that the phenotypes that we measure are the best representation of the breeding values. Another assumption is that the phenotypes are corrupted by a complex function of soil, microclimate, and genetics. This is likely the case. However, the problem with this is that we really don't have the data to fit this model because we do not have meter by meter soil data in our vineyards. So one could use the location of the vines as a proxy for that effect. And so that's essentially what we're going to do here. We're again going to use a mixed model. Here, the response will be corrupted phenotypes, or what I'll call corrupted phenotypes, which is simply the phenotypes that are measured in the field. And that's going to be explained by some spatial effects. And the noise will be our uncorrupted phenotypes. And so the spatial effects will be controlled by this L matrix. It's somewhat similar to a genetic relationship matrix. Instead of this matrix being a function of SNPs, it's going to be a function of the location of the vines in the field. And so there's a number of ways you can encode this, but I'll just talk about two. One is called a moving average covariance model, and the other is an autoregressive. And they're commonly used in time series analysis. So to explain these models, I'll have a really simplified example where I have 4 vines in a row, and I'm going to see how or what covariances these various models give. So the simplest one is called a moving average one model, where you're only modeling the covariance between adjacent vines. And this, you could see the encoding of this, of the L matrix here, where this A, which is a value between 0 and 1, is kind of the covariance between the first and second vine, and goes down the diagonal. So this can be represented here. So a moving average 2 model is a little more complex, where neighboring vines are explained by A plus A times B. Again, B is a value between 0 and 1 that we're going to optimize for. And B, the covariance between the first and third vine is a value of B. And here's the corresponding covariance matrix. An autoregressive one is simply saying that there's an exponential decline or an exponential decay of covariance as you go further along. So neighboring vines have covariance of A, then the next one then between the first and third is A squared, and between the first and fourth is A cubed. And so because A is between 0 and 1, as you go further along, you're going to get close. You're going to have covariances closer to 0. So then you have an autoregressive 2, which is a little more complicated. Essentially, the easiest way to think of it is as combining 2 autoregressive 1 covariances. And so again, you have this B parameter now. However, if you set B equal to 0, it's going to revert back to an AR1 covariance. And so here, this L1 is simply this value, and L0 is 1. And so the covariances are related to the previous covariances. So to evaluate how well these models perform, I have a data set which is again, an F1 interspecific cross similar to the previous crosses that I've explained. There's 6 rows here and 110 vines within a row. I'm only going to model the covariances within a row because of the small number of rows that we have. There's 475 vines that have been phenotyped for powdery mildew on a 1 to 5 scale in 2002, and 350 of them have genetic data. So how I'm going to do this evaluation is I'm going to fit that mixed model that I explained previously. And with the uncorrupted phenotypes, I'm going to do a QTL analysis. And I'm also going to leave 10% out cross validation. So what that means, I'm going to use 10%, I'm going to use 90% of the genotypes that have genetic, or phenotypes that have genetic data as in this model. And so this model is essentially Divilov, which is, again, another mixed model where similarly how we had that spatial covariance matrix, we now just have the genetic covariance matrix as explained by Van Radden. And then we predict the test genotypes with the parameters estimated from that model. And we're going to compute an accuracy score, which is the Pearson correlation between the predicted and true uncorrupted or corrupted phenotypes. So looking at the results, the spatially unadjusted and adjusted phenotypes detected the same 3 QTL in the same locations. Looking at the genome-wide prediction results, the accuracies, there was an increase whether used an MA1 or any moving average or autoregressive covariance function. However, the standard deviations were quite high, and so this increase was not statistically significant. Looking at how well the covariance matrix fit the data, an AR1 and an AR2 here fit the best, but they actually fit the same because the AR2 reverted to an AR1 because the B parameter was set to 0 during optimization of, in this case, the log marginal likelihood, or this is what REML optimizes, or what REML is. So I have 2 more data sets. In this case, I do not have genetic data, so I'm just looking at how well they fit, how well the covariance matrices fit the data. So again, they're both F1 interspecific crosses. The first one has 4 rows, 76 binds per row. Again, only modeling covariances within rows. About 300 vines were genotyped, again, for powdery mildew in 2017. The second vineyard had 3 rows, 61 vines within rows. About 200 vines, second-year vines, were phenotyped for height. And so these vines were never trimmed. And so we could phenotype for height in this case. So the results here is that AR2 covariance functions fit the best because they had the greatest log marginal likelihood. So to summarize, the 2002 powdery mildew data set did not show a significant benefit to adjust for phenotypes. However, it would be good to have some more vineyard data where we could look into this, especially one where the standard deviation isn't so high. And that the AR processes usually fit vineyard data better than moving average processes. So just going back to our overview of grapevine breeding, where this research fits in, the high throughput phenotyping using computer vision helps us obtain faster phenotypes for our F1 families. And this is where we get our markers that we use for marker-assisted selection. And accounting for spatial effects could possibly lead to more representative, accurate QTL or ratings in the field. And the Bayesian networks, for example, could suggest what phenotypes we should generally breed for if we want disease resistance. I'd like to acknowledge Bruce Reich for giving me the independence to conduct the research that interested me, Lance Gale Davidson and Michael Gore for helping me flesh out my research and for being on my committee, and everyone else here for helping me out with my research. And I'd like to thank my funding sources, Bayesian, which was funded by the USDA NIFA grant, and the Charles R. Bullis Plant Hybridization Endowment, and the Michael Nolan Endowment Fund. Thank you, and I'll take any questions.
Let me ask one question, Konstantin. Do you think the work that you did on the Bayesian network analysis, do you think that could have been applied to many past studies in QTL analyses in vitis, or do you see that applicable to most QTL analyses?
No, because in most cases we only had, or I feel most studies only had one phenotype, and I don't think there's a benefit when there's just a single phenotype. I think the model will be very similar to what you get with that single phenotype QTL analysis.
Did you try using multiple rows instead of just within row spatial adjustment?
Accounting for the 6.
So you said you had multiple rows.
Yeah.
Did you try combining the information in those rows in your spatial analysis?
Yeah, and it actually decreased the, again, it wasn't significant just because of the high standard deviation. But I think the issue there is that we would have to, so I guess I didn't mention this, is that the space between vines within a row is different than the space between rows. And so we're going to need different A and B parameters when we fit between row covariances. And since we only have 6 vines, or sorry, because we only have 6 rows, those parameters are likely not to be like, yes, really well estimated, which I feel is what happened. Yeah.
How common are those trichomes on the vitis species that are used or looked at for O-mycelial fungal resistance?
Yeah, so technically, Vitis vinifera has leaf trichomes, but they're practically not seen as shown in the leaf disc image that I've shown. But those really dense leaf trichomes, they are never present in Vitis vinifera. They're present in certain Vitis species like Cinerea and Labrusca, just to name 2. I think there it's the most common. So it's not all wild species have these dense leaf trichomes, but just some do.
So the interesting thing about the trichomes is that there have been some other studies just on the inheritance of trichomes too. I think some some of your analyses pointed to the inheritance patterns of trichomes. And they're also related to the ecological niche in which certain types of predatory mites exist on great leaves. So you can actually encourage certain predatory mites to live on great leaves just by breeding for higher trichome densities, or domatia densities.
Okay, well, do you have a question? Oh, okay. Well, if there's no more questions, thank you for coming to my seminar.
This has been a production of Cornell University, on the web at cornell.edu.